Hi everyone, my name is Dylan, and today I will be starting a new video series where in each video I go through a different H2 bio practice paper covering a different topic each time. So this video is created to bridge that learning gap by really showing you guys the thought and reasoning processes involved in formulating your answer for each H2 bio question. At this point, I should also put forth a disclaimer that the questions I'm using in this video series are not past year A-level questions, and they are also not questions from past year school prelim or promo papers. The reason for this is that I believe there are actually some copyright restrictions placed on those papers, so if I were to just showcase and publicize them online for everyone to see, I could potentially be in deep legal trouble. So instead, what I've done is I've actually created my own set of mock questions based off of my 2 years of experience learning H2 Bio and also as a volunteer tutor for H2 Bio, which has exposed me to hundreds and hundreds of TYS and prelim papers. So I think my papers should be a pretty good reflection of what the actual A-level standard is like. So today's video will be covering the topic of biomolecules. And we've really got the full range of question types covered. There will be basic knowledge questions, data analysis questions, application questions, and even some practical questions. Additionally, I just wanted to add here that if you guys want to make the most use of this video, you should definitely make sure to brush up on both your knowledge and understanding of the topic that I'm going through, just so that you can go on to apply that knowledge into the more novel contexts that I introduce in my questions. I very highly recommend that you actually pause the video before I start to go through the questions, just so that you can have a look at the questions yourself and attempt them under timed conditions if possible. So without further ado, let's get to the questions.
So question 1 says that statements A to E refer to various biological molecules. And for each statement, we basically have to identify the term that most appropriately matches the description. So let's look at what statement A says. Statement A is the reagent used to test for the presence of reducing sugars. Okay, so this is a very standard test that you may encounter a lot when you're doing your biopracticals. And what they're talking about has to be the Benedict's test. So just take note that the Benedict's test is specifically used to test for the presence of reducing sugars. If you are testing for the presence of non-reducing sugars, then you have to include an acid hydrolysis step before the Benedict's test so that you can break down the non-reducing sugar into monosaccharides that are reducing sugars themselves. For statement B, we have an unbranched polymer consisting of only alpha glucose monomers. So there are two parts which are important. The first part is how the polymer consists of only alpha glucose monomers. So we can immediately rule out cellulose because cellulose contains beta glucose monomers. And another thing is that the polymer is unbranched. So we can rule out amylopectin and glycogen because both of those molecules are branched. So that only leaves us with amylose because amylose is the only unbranched polymer we know in the H2 syllabus that consists of purely alpha glucose monomers. So amylose is the right answer. C says the molecules formed from a condensation reaction between two glucose molecules. So take note that condensation reaction should not be confused with hydrolysis. In fact, condensation is the opposite of hydrolysis because in hydrolysis, you are using one molecule of water to split a larger molecule into two smaller molecules. Whereas in a condensation reaction, you are forming a larger molecule by joining two smaller molecules together and in the process releasing a molecule of water as well, hence the name condensation. So in this case, we know that water is one of the molecules formed, and the other molecule formed is basically the disaccharide that is composed of two glucose subunits, and that would be maltose. So maltose and water would be the right answer. Let's move on to statement D. The molecules produced when a polypeptide chain is hydrolyzed. Okay, so this ties back to what I said earlier about how a hydrolysis reaction is basically a reaction where you use a molecule of water to split a larger molecule into smaller subunits. So in this case, a polypeptide chain would be composed of many amino acid subunits. So the answer is just amino acids. Take note that water molecule is not part of the answer this time because water is the molecule that we add to the reaction. We do not produce water by hydrolysis. We only produce water through condensation. So finally, we have the name of the bonds broken when a triglyceride is hydrolyzed. You need to know that a triglyceride is composed of a glycerol backbone joined to 3 fatty acid chains via ester bonds. So the name of the bonds broken would be ester bonds. So now let's move on to the next question, which is an application question on carbohydrates. The question says that glycogen branching enzyme and glycogen synthase are enzymes that catalyze the synthesis of glycogen. Part A asks us to name the bonds present in glycogen. So glycogen, we need to know that it is a branched molecule, and it is a molecule made up of alpha glucose monomers, as I mentioned earlier. So the bonds present would be alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. Okay, part B is slightly more difficult and interesting. It asks us to suggest and explain why glycogen branching enzymes are needed for glycogen synthesis when glycogen synthase is already present. So take note of this important criterion mentioned in the question. The question isn't asking you to give a generic explanation of why enzymes are needed to catalyze the synthesis of glycogen, because the question specifically asked you to consider the fact that glycogen synthase is already present. So why do I still need a different type of enzyme to synthesize glycogen, right? So therefore, it is important that your explanation is specific to how glycogen branching enzymes operate. So the clue is actually in the name of the enzyme itself, glycogen branching enzyme. And branching suggests that the enzyme specifically catalyzes the formation of alpha 1,6 glycosidic bonds because these bonds are what forms the branch points in glycogen. So it's very likely that what is happening here is that glycogen synthase is not able to catalyze the synthesis of these alpha 1,6 glycosidic bonds, leaving glycogen branching enzymes to do the job instead. So of course that's not the end to the answer because the question is worth 3 marks, and it also asks us to suggest and explain why the enzymes are needed. So rather than just stop at saying that the branching enzymes help with the formation of branches on glycogen, it would be good to explain why glycogen requires branches in the first place. So that actually ties back down to our theory knowledge about the adaptations of glycogen to function as a good storage molecule. So what I will first talk about is the fact that it synthesizes alpha 1,6 glycosidic bonds and forms the branches of glycogen. And afterwards, you can go on to mention why the branches are important. Firstly, the branches help to increase the amount of free ends available for faster hydrolysis by enzymes. So this allows glycogen to release large amounts of glucose molecules efficiently when it needs to get hydrolyzed by enzymes, for example, during respiration. And the third point, you can talk about how increased branching actually helps glycogen become more compact. And this is good because it allows more glycogen to be stored in the cell per unit volume. So that will be our 3 points for this question. Okay, so let's move on to question 3, which is our practical question for biomolecules. So usually for H2 Bio, the practical questions that test on biomolecules are most of the time food test questions. So 3 part A, a student wanted to find out whether starch was present in a sample of plant tissue. So they want you to describe the food test for the presence of starch. And basically, this is a very standard knowledge question. You need to know that iodine is used to test for starch. But here's the part where many students make a mistake, because they often say that, oh, you just add iodine to the plant tissue, and if the plant tissue turns blue-black, then starch is present. So this answer is problematic because in reality, the food test for starch doesn't just use pure iodine. It uses iodine specifically in potassium iodide solution. So it is compulsory that you mention this in your answer. Iodine in potassium iodide solution. And generally, when you want to write the procedure for a food test down, you need to make sure that you talk about both the positive observation when starch is present and the negative observation when starch is not present. So the basic formula for writing this kind of food test is to first describe what the test is. So in this case, once again, I'm adding a few drops of iodine in potassium iodide solution to the sample. And then usually what I do is I talk about what happens if starch is not present. So if starch is not present, then the sample would turn yellow-brown, which is just the color of the iodine solution. And if starch is present, which is the positive result, then the sample should turn blue-black, because this blue-black color is actually the complex that iodine forms with starch. So part B is another very similar question, except this time round, we are describing the test for a protein instead of for starch. So the test for a protein is basically the Biuret's test. And once again, the formula is to first start off by describing what the Biuret test is, and then go on to describe what are the results if proteins are absent, and the corresponding result if proteins are present. So here is the answer for this question. So I'm just going to add a quick note here that the Biuret test can only work for testing proteins. It does not test for the presence of amino acids. And the reason for this is that the Biuret's test detects the presence of peptide bonds. It does not detect the presence of amino acids. So just take note, if you encounter an MCQ that asks you whether the amino acids can be tested for by Biuret's test, the answer is a straight up no. So we have this final part C, which tells us that a student was told about a particular solution which may or may not contain a certain disaccharide, which includes your maltose, sucrose, and your lactose. So the question asks you to suggest how they could verify that the solution contained sucrose. You cannot just use Benedict's test directly to test for the presence of sucrose, because Benedict's test specifically only tests for the presence of reducing sugars. Even if non-reducing sugars such as sucrose are present, the Benedict's test would give a negative result. So of course, it can be very tempting to just give the answer as the typical acid hydrolysis followed by Benedict's test reaction. However, if you do that, there is a problem that may occur, because the question states that the solution may contain a certain disaccharide, which means not just sucrose, but also lactose and also your maltose. So think about what happens if you use the acid hydrolysis on maltose or lactose. Maltose or lactose will also get hydrolyzed by the acid to form monosaccharides, which are all reducing sugars. So even if you do that, your Benedict's test would also give the same result as that for sucrose, and therefore it is insufficient to just use the acid hydrolysis plus Benedict's test to test for the presence of sucrose, because you haven't ruled out the possibility that lactose and maltose are present as well. So instead, what you need to do is you need to do the Benedict's test twice. First, you do the regular Benedict's test to make sure that maltose and lactose are not present. Because if maltose and lactose are present, then this would mean that the first round of Benedict's test should already give you a positive result. But if the solution remains blue, then that means that either sucrose is present or there is no disaccharide present at all. So once you perform the first Benedict's test and you have make sure that the solution remains blue. You then can go on to perform the acid hydrolysis step, followed by the second round of Benedict's test to see whether a positive result is obtained this time. So if only sucrose is present, then only the second Benedict's test will produce positive results. And the positive result in this case would be the formation of a brick red precipitate in the solution. So now we are moving on to a data analysis and application question for proteins. Question 4 says, the diagram below shows the oxygen dissociation curves for fetal hemoglobin, otherwise known as HbF, and adult hemoglobin, otherwise known as HbA. So part A asks you to describe and explain the graph shape for HbA, which is adult hemoglobin. So typically, the main thing you need to observe in this graph is the fact that adult hemoglobin's curve has a sigmoidal shape. And this sigmoidal shape is linked to the fact that HbA displays subunit cooperativity, which I will go on to elaborate in my answer later. And also for described questions, you want to make sure that you quote the data given. So that is exactly what we are going to do to answer this part A question. So describing the graph shape, we just start off by talking about how the graph shape is sigmoidal. And once again, that is linked to the allosteric nature of hemoglobin, as I mentioned earlier. So as the partial pressure of oxygen increases from 0 to 26.8, the oxygen saturation of HbA increases exponentially from 0 to 50%. So the word exponentially is important because that is what I'm directly seeing from the graph. The fact that there is an increasing steepness of the gradient as more and more oxygen binds to hemoglobin contributes to the formation of this sigmoidal shape. But the exponential increase is not the only part of the sigmoidal shape. You need to also go on to mention that as the partial pressure of oxygen further increases from 26.8 to 120, the oxygen saturation of hemoglobin increases, but this time with decreasing steepness from 50 to 100%. So this decreasing steepness is the second half of like the S shape you see in the curve. And you can also go on to make special mention that the oxygen saturation begins to plateau at a partial pressure of around 80. Because that's not all for our answer. So far I've only described the graph shape. I haven't really applied any of my theory knowledge to explain the graph shape. So in my explanation, I'm just going to make reference to how there is subunit cooperativity in hemoglobin. So what I'm going to talk about is how hemoglobin is an allosteric protein with 4 subunits that each bind one oxygen molecule at the heme group. So when one oxygen binds to a heme group, it actually induces a favorable conformational change in hemoglobin such that the binding of oxygen to other heme groups of hemoglobin is facilitated. And this increases the affinity of the other hemoglobin subunits for binding oxygen molecules. And that is basically what subunit cooperativity or cooperative binding represents. Okay, so for B part 1, we have something a bit more interesting. They give us a new concept known as this P50 value. And they define it for us as the partial pressure of oxygen that results in 50% saturation of hemoglobin. And the question is, suggest what the P50 value indicates about the binding affinity of hemoglobin to oxygen. Alright, so if we look back at our graph, we notice that there is something interesting because, like what they said, the P50 value is the partial pressure of oxygen, which means the x-axis value, which gives us a 50% saturation of hemoglobin with oxygen. And we can actually already see it being marked out on the graph right here. So what could this actually mean? So you can think about it logically, because if the P50 value is lower for the particular hemoglobin molecule, it means that you need less amount of oxygen to achieve 50% saturation of hemoglobin. So if you think about it this way, it means that the lower the P50 value, the stronger the binding affinity for the hemoglobin molecule to oxygen, because now you need to bind to less molecules to achieve the same saturation, which means you essentially are binding oxygen stronger. So we can just answer the question with this insight in mind. We mentioned that the lower the P50 value, the greater binding affinity of hemoglobin for oxygen. Okay, so for B part 2, there is another interesting context given to us. It gives us information about how a developing fetus is unable to obtain oxygen from the atmosphere and can only receive oxygen by diffusion from the mother's blood through the placenta. So they want us to use our answer for B part 1 to explain the difference in P50 values between HbF and HbA. So for B part 1, we already established that the lower P50 value there is, the greater the binding affinity of hemoglobin for oxygen. So at this point, we can just check back at our graph and compare the P50 values of HbF and HbA. And we notice that HbF actually has a smaller P50 value of 19 compared to the P50 value of HbA, which is 26.8. So we can first start off by quoting this particular piece of information from the graph, that the P50 value of HbF is actually lower than that of HbA. So using our answer for B part 1, that must mean that HbF must somehow bind to oxygen with higher affinity than HbA. But of course, we cannot stop our answer here because that's still not explaining why HbF needs to bind oxygen with higher affinity than HbA. And that's when we need to use the information given to us in B part 2. So in B part 2, they mentioned that the fetus needs to obtain oxygen by diffusion from the mother's blood. So you need oxygen to somehow be released from the HbA present in the mother's blood so that it can bind HbF. And that is why you need HbF to bind oxygen with higher affinity, because it effectively creates this affinity gradient, which draws blood away from HbA in the mother's blood to bind HbF in the fetal blood. And this ensures that the fetal blood can supply sufficient oxygen for the growth of the fetus and so on. So that is why HbF needs to have a lower P50 value than HbA. Okay, so we are left with one final part, which is part C. And once again, we are presented with a new piece of information that we need to apply our knowledge to. So it says that just like hemoglobin, myoglobin is a protein that binds oxygen, and it's found in the cardiac and skeletal muscle tissue of humans. Unlike hemoglobin, myoglobin does not display subunit cooperativity. And basically, they just want us to suggest one difference between the structures of both proteins. So to answer this question, we just need to showcase our understanding of what subunit cooperativity is. So if I just scroll back up to my answer for part A, I mentioned that subunit cooperativity basically involves this concept of allostericity, where you need multiple subunits that can each bind one oxygen molecule. So it is very likely that because myoglobin does not display subunit cooperativity, myoglobin is not in fact an allosteric protein, meaning it only has one polypeptide chain. And that is why the concept of subunit cooperativity doesn't really apply to myoglobin, since myoglobin only has one subunit to work with in the first place. So that is the difference that we can talk about how myoglobin has one polypeptide chain versus 4 polypeptide chains that are present in hemoglobin. That brings us to the last question, which is really just a basic knowledge question on the topic of lipids. So question 5 asks us to compare the structures of triglyceride and phospholipid, which are both different types of lipid molecules. So the key takeaway for this question is on this keyword, compare. So even though this question is 4 marks, it is not sufficient to just give 4 differences or 4 similarities, because the A-level document specifically states that compare as a question term means that you have to give both similarities and differences. And it is ideal that you give an equal weight to both, meaning you should have 2 points on similarities and 2 points on differences. Also, a very important thing to note is that when you are giving differences in the A-level H2 bio questions, you should always compare the two items point to point. So what I mean by this is you should compare the two things using a common criterion. So let me illustrate this using my answer. So I'll start off by giving one difference, and that is the fact that phospholipids have a phosphate group, while triglycerides do not have a phosphate group. So over here, you can clearly see that this is a point-to-point comparison because I have a common criterion here, which is the presence of a phosphate group. And I'm just saying that phospholipids have it, whereas triglycerides do not. So for the next difference, I'm talking about how phospholipids have 2 fatty acid chains in their structure, while triglycerides have 3 fatty acid chains. So once again, I'm using a very point-to-point comparison here because the common criterion I'm using here is the amount of fatty acid chains, and I'm directly stating and comparing the amount for the phospholipids and the amount for triglycerides. So it's a very clear-cut difference right here. So now that I have my two differences, it is time that I also start to talk about the similarities to address the compare part. So one similarity I can talk of is about the fact that both phospholipids and triglycerides have ester bonds, because ester bonds are the hallmark of lipid molecules. They are what join the glycerol backbone to the fatty acid chains. And along this same line of thought, I can talk about how both triglycerides and phospholipids have a glycerol backbone. And that will be our final similarity for this question.
Alright, so that will be the end of our practice paper. If you enjoyed the walkthrough today, please like the video and share it with others. In the meantime, I'll see you guys in the next video. Goodbye!